Hello and welcome to this week's What Were They Thinking? To begin with, we have some interesting news about the possibility of a natural or innate degree of immunity to the CCP virus. As you may or may not know, most people have a diverse immune system, and that this is selected for over time. Gradually, as you get older, the body removes more and more unused immune cells. When you are younger, primarily between the late teens and into your middle age, you retain a large and diverse set of cells. These are able to recognize and respond to a large and wide range of pathogens. This then plays into the study coming out of the journal Cell that showed that some people have helper T cells that are able and capable of responding to the CCP virus. This is a phenomena called cross-reactivity, where the ability to deal with one pathogen imparts a degree of ability to respond to another pathogen. This is often useful, especially when looking at things like the influenza vaccine. In this case, the cross-reactivity has imparted a greater advantage in immunity to the CCP virus. Unfortunately, their study was on a very small scale. We're talking 20 people in the experimental group and 20 in the control group. But the fundamental basis of this experiment is sound and would be rational to think could be expanded to the wider population. But as is a trope within research, more research is required. Next, we have an article coming out of the conversation. This is all about how we can expect to see a second wave, much like we did with the Spanish flu and even the swine flu incidents of 2009-2010. The article from the conversation shows both the rationale and method behind this second wave, primarily how they use a designation of R, which is how infectious the disease is and how many new cases come from each person who has been infected. It continues to show how in many countries you have an above 2 ratio, which is for every 1 infection, there are 2 new infections generated, and that after the lockdown and restrictions in many countries, this has come down below 1. That means for every new infection, they are able to create less than 1 new infection thereafter. That means 2 infected people might lead to 1 more infected person, and so on. This is important to understand when you're looking at why diseases become stuck and entrenched in society. Consider influenza. It moves from the northern hemisphere to the southern hemisphere, and based on this, we can see that there's a continuing motion of infection, recovery, and reinfection. The conversation article provides a lot more detail and is really interesting if you want to look at how epidemiologists model events like this, and even extrapolate it out to events like the flu, viruses, and other pandemics. Good information, well presented, have a look. While looking at that article from the conversation, bear in mind the following. Some research coming out of Hong Kong shows that the majority of cases are caused by just 20% of the infected individuals. That is, 20% of cases infect 80% of all following cases. It's very similar to the statistics you find for many naturally occurring phenomena. There's also a theory that at least 70% of cases altogether don't pass on an infection at all. It's believed that there's likely to be some sort of super spreader involved, and that's what they consider this 20%, a fractional amount responsible for spreading the disease to everyone else. The article itself is only a preprint, so it should be taken with a grain of salt. But it's worth thinking about based on what we've seen so far, that many people don't seem to be experiencing significant symptoms. There's a particular group of individuals who are very susceptible, and even within the group that is not susceptible, we're seeing unusual phenomena. Next we have a bit of an interesting story, and it's part of a larger series of issues at the moment. It relates to hydroxychloroquine. Last week we mentioned a large-scale study that seemed to have found no positive benefit and possibly a negative. That was an observational study and lacked a clinical arm. Now we're seeing clinical experiments, or clinical trials might be a more appropriate description. The one in question involved 821 patients across the United States of America and Canada. What they found was that the benefit was comparable to giving patients a placebo. Now, that's an important thing to bear in mind. A treatment arm and an experimental arm with a relatively large cohort. The study itself had some big flaws. First, the majority of patients were deemed to be a high-risk group. That is, they had been in contact with people without wearing protective equipment. That's something to bear in mind. Mind you, the people they had been in contact with needed to have had a confirmed case of the CCP virus, so there is at least some established risk there. Next, they were either given the hydroxychloroquine or they were not. They were then monitored over 2 weeks. Yet another weakness is that they were either given a laboratory test or they were watched for clinical signs of infection. What they found was that 49 of the 414 given the hydroxychloroquine became ill, versus 58 of the 407 who received the placebo. That is an 11.8 versus a 14.2% difference. Something that comes out at a little under 3%. In the coming weeks and months, the US is expected to begin stockpiling millions of doses of the various CCP virus vaccines that have been made by companies. This has been seen as a good sign by the director of the National Institute of Allergy and Infectious Disease. The argument behind it is that with this large stockpile of vaccines, they could look at saving hundreds of lives and possibly treating hundreds of millions of people. The problem behind all of this is that they're all early stage clinical trials that they're going to be testing these people with. Stage 3 trials to be precise. Stage 3 trials are the earliest stage before a drug is given approval. After this you have stage 4, in which you have large-scale trials and observational studies, but generally speaking, at that point in time, a drug is being given approval, as a pharmaceutical company can't afford to invest that much money, time, and resources. Bear in mind that development of a vaccine treatment of any kind normally takes years, even decades, and these are being brought to a phase 3 trial within 6 months of the big companies starting their investigation. This is one reason why Dr. Fauci is immediately wary that these drugs may not work as intended, that we may not get a viable vaccine, but he has stated that he's optimistic that something will work by the end of the year. It's this issue with statistics that we need to look back on something from last week. We mentioned an article that had been published about the benefits of hydroxychloroquine, or more accurately, the lack of benefit and possible negative effects. A lot of this research has been based on an emerging controversy around decisions made on that data set and others. Much of the research was based on a company called Surgisphere, who claims to have a variety of sources where they work with hospitals, but they have a very sordid history. As a consequence, looking at the current body of evidence and the decisions made based on it, there's a lot of uncertainty and a lot of concern. There's a lot of criticism about the company and some of the employees, particularly that there's a sci-fi writer, an adult content model, and more. Now, there's no reason to criticize individuals for their history or their chosen career path if they are able to do their job. But the concern is that people think they may not be capable of performing the function they claim to have performed. This has led to both The Lancet and the New England Journal of Medicine issuing concerns in their journals about the articles and data. What is more disconcerting with this particular development is not so much the nature of the fraud, but how it was discovered. It appears The Guardian in Australia, of all outlets, is the one to have found it, and they provide a full outline of the concerns they raised, some of which is that the number of patients who have died didn't match with the timeline given, that after they contacted a variety of hospitals, they denied any involvement in such data collection and providing it to another party, and there are a variety of other problems that they have raised in their articles, and they're worth a read, if only for this. Moving now away from the whole nonsense that is the CCP virus, we can look at some interesting science. In this case, there's a curious example of red blood cells being developed that could perform better than real blood cells. Understand, the human red blood cell, or erythrocyte, is capable of pushing a lot of oxygen around the body and then bringing the wasteful CO2 back and expelling it via the lungs. This is extremely efficient considering what it is, and that each cell fundamentally lasts 180 days from when it is first released into the body and then destroyed. These cells, if they really are as effective as claimed, would be incredibly useful, especially when it comes to delivering drugs to specific areas. The red blood cell is a very basic cell. It contains the hemoglobin, the oxygen, and enough energy within it to be able to perform its function, but it lacks a nucleus. That means it is a one, done, and disposable cell type. In theory, in the body, it could be incredibly useful for delivering medicines for that reason. It's relatively non-reactive, it's very flexible, and it can move to many locations that larger cells don't have the ability to access. Another similar development involving synthetic organs, treatments, and more is that in which human livers were grown in a lab and then transplanted into rats. For years we've been growing various organs, parts of bodies, and more on the backs of mice. This has been a relatively effective process, but it's generally not very good for the animal, and it hasn't represented a viable pathway for developing these for transfer to humans. This process involved growing the organ effectively in a test tube, then transferring it across to the animal. If the tiny organ being grown for the rat could be sized to that of a human, it is viable that we could have a means of resolving some of our ongoing issues with a lack of transplantable organs, especially in large countries that lack a donor base. By itself, the discovery is a great proof of concept, but it's how they did it that is really impressive. To begin with, the livers were not grown using liver cells. Instead, they used human skin cells that were donated. Then they used a process of various signals to induce the cell back into a pluripotent stem cell state. That is, before the cells differentiated to become hepatocytes. The important thing with pluripotent stem cells, and this goes back to some of the controversy from many years ago about stem cells, is they can turn into anything. For many, many years, they were only able to be sourced from developing fetuses and similar, where they had yet to diversify. In later years, we realized that there are pluripotent stem cells available in the adult, and if not pluripotent, then at least only partially diversified cells, and so you could, in theory, take these cells and backwards engineer them and turn them back into pluripotent stem cells, or continue to induce changes in them to continue the avenue they were going down, but choosing which particular path they would go if there were multiple options available. The way they did it is further greatly interesting. They took the cells they had created, and unlike the normal 2 years of growth for a liver, they were able to transfer the cells to a rat liver that had had all the cells stripped from it. These cells were then replaced with the seeded cells from the human skin, which had been turned into pluripotent stem cells. It was then given the appropriate signals to turn into the liver cells over a period of weeks rather than years. The study was not without its flaws though, and there was evidence of poor blood flow, thrombosis, ischemia, and other issues with getting the grafted tissue to connect to the vascular system of the rats. Now, in a human, we may not encounter the same problem, but we also might be facing issues with getting the graft to take properly. However, as a proof of concept, this is a very big milestone. In some astronomical news, you may have noticed over the last day or so that the moon has been incredibly large and bright. This all owes to the fact we're about to enter penumbral, that being a period where the moon is bright and eventually is eclipsed. If you didn't get the chance to see it, there are generally a variety of sources of information out there that you can go to watch the transition of the moon and the eventual eclipse, which should have lasted a little over 3 hours. In other spacefaring news, we have confirmation that SpaceX has been able to safely deliver the astronauts who went into space on Tuesday. Their launch marks the return of American soil-based launches of rockets into space itself, something we haven't seen for years, and where America has been relying more and more on Russia. SpaceX needed to do this to mark its capabilities and confirm that it is capable of achieving NASA's goals using its own proprietary rockets and technology. It was competing with a number of other companies, of which only one has actually been able to produce rockets and enter into a contract with NASA. While talking about the SpaceX launch, there's actually an interesting aspect to it, beyond the fact that we have successfully relaunched people into space from America using a privately made rocket. That is, the bizarre image you might see cropping up here and there. There have been some conspiracy theories put forward about it, such as it's a mistake by the crew and it's all fake, but in fact, the small, plush toy dinosaur you see in these images is both a tradition and an interesting measure. The image you see is that of a small sequined toy dinosaur, and it's in the cockpit. The reason it's there is simple. It's an indicator of when the humans on board have entered space, as it is so light that it will begin to float. This is a convenient, relatively safe, and effective tradition that gives them a measure. This is also why, on occasion, you'll find them called zero-gravity indicators. Next we have a disturbing story coming out of Australia, although there are very few that aren't. Researchers looking into fish in the area have found that there's a disconcerting number of live eels being found in the fish. It's not just any old eel though, it's a specific one that has some curious physical anomalies. That's why when we're looking at this, the images may be disconcerting for you. It's a species of eel known as the burrowing snake eel, and among the tools available to it is a rather hard, pointy tail. The reason we bring this up is that it seems a number of these eels have been eaten whole and alive by large predatory fish. Having been consumed this way, they've been trying to find their way out, and being a burrowing eel, you can imagine what their only real solution is. What we have found is that many of these fish either become dormant or don't survive, and the eel becomes mummified within them. The researchers involved seem to believe that this is a relatively rare phenomena, and that they're probably encountering the unfortunate eels that have survived, those who are a very small portion of the predatory fish's diet, and that they need a larger sample size to understand what is actually happening, and if further investigation will be able to elucidate that answer. Finally, we have an interesting development in sodium, or at least sodium chloride, otherwise known as table salt. Normally when this forms into a crystal, it creates a cubic shape. There's nothing exciting about this, we've understood the format for years, and ultimately it's not that useful, other than on food. In this case, they're not forming it into a cube, but through modeling and then development of a methodology, they are able to create a hexagonal structure using the sodium chloride. Unlike the cubic shape of normal salt, this was done on a 2D format, and it had to be done on a 2D format at a size of micrometers. They were able to make their hexagonal salt at just 6 nanometers thick. What they found was if they tried to make it any thicker than that, it would begin to turn back into a cube, and that would not be particularly useful. The reason they are after making it into a hexagon is simply this. There's a variety of electronic devices that currently rely on boron-based hexagonal crystals. These crystals aren't as stable as what you would get if you were to use the sodium chloride crystal. This means that if there's any luck going forwards and they can create it at an economical scale, we could look at having some electronic devices performing at a far better level than now. In particular, electric cars are a big target. That's the last of the stories we have for you this week. Thank you for watching. If you found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions you may have below.